Hi everybody, I'm David Broussard from the first year of Maris, and I'm happy to present you my immersion project at the Institute for Research on Cancer and Aging in Nice. The IRCAN is based in the north of Nice and shares a building with the medical faculty of the University Côte d'Azur. The institute focuses on biomedical research in the field of cancer and aging. From fundamental research to the medical application, different models are used to understand this complex topic. In the Eric Rettinger team, where I was able to do my immersion project, the focus lies on the Cnidarian model Nematostella vectensis. This sea anemone is of special interest since it possesses high regenerative capacities. Post-amputation induced by bisection underneath the pharynx, the sea anemone is able to regenerate its missing body part within a week. The first step is the wound healing, followed by a fusion of the mesenteries in contact with the amputation site. After 7 days, the animal is regenerated with a fully formed pharynx and a mouth opening. This is not only cool but also very interesting, since Nematostella has some important structures and cell types shared with bilaterians. A surprising abundance of human disease genes are present in the starlit sea anemone. But there's no specific sign of aging or cancer. Why though? To answer this question, it is important to learn more about the organism's physiology and response to certain stressors. A small piece of the puzzle is its response to extracellular pH variation, observing the whole organism, the tissue, and the cells. We performed a lot of different pH experiments, long and short term, in a range of pH 2 to pH 14. An interesting finding is concerning the regenerative capability of Nematostella in various pH conditions. We were able to define regeneration limits for adults and juveniles. The limit for adults is reached with pH 4 and pH 10, and for juveniles with pH 5 and pH 9. A 100% regeneration could only be observed in the range of pH 6 to pH 9. Besides that, we were able to observe an unexpected resistance towards tissue degradation of bisected animals in extreme pH conditions. Bisected animals, so animals that have already been stressed, seem to tolerate extreme pH conditions longer than non-stressed animals. The non-stressed tissue disintegrates minutes after being placed in the solution. But we've done much more. We also analyzed the presence of micronuclei in the different pH conditions to see if there's any sign of DNA damage, with the help of a nuclear staining called DAPI, a blue fluorescent stain that binds to adenine-thymine regions in the DNA. We also had a look at the muscle network, the mitochondrial activity, reactive oxygen species, the cell proliferation during regeneration, and many more. All of this needs to be explored more in depth, but this preliminary data already helps us to understand some of the physiological responses and especially limits of Nematostella vectensis that might be applicable to other cnidarians and species with high regenerative capacities. If you're interested in all of these findings, please make sure to not miss my talk in December, and don't forget to subscribe to the Maris YouTube channel to get all the new updates. I would like to give a special thank you to Aldina Miel and Eric Rettinger for their help, their expertise, and their encouragement during my project. See you soon, guys, and have a wonderful summer.